Hello and welcome. Welcome to this video. Welcome to my channel if you're here for the first time. This video is a little unusual for my channel. My channel is mostly about bioinformatics, genomics, and related topics. This video is a response to a video by another person, a Dr. John Campbell, who put out a video with what I believe is some serious misinformation comparing the new Pfizer antiviral compound to ivermectin for treatment of COVID-19. So I'm responding in this video to address the misinformation in that video. If you are interested and you want to skip around, so if you've seen his video, you may want to skip around, and there'll be links to chapters below. There'll also be links to all the literature referenced below. And I encourage you to leave comments and let me know your questions and your input on this. Okay, so let's get started. So let me start by just addressing the question, why this video? Why do this video at all? Well, Dr. John Campbell has had a huge influence. He's been making videos for a long time. He makes them very well. And he's developed an increasing following during the COVID-19 pandemic through his YouTube channel. He has something like 1.3 or more million followers. He puts out videos every couple of days, so he really draws a lot of following. In that, his clinical background and scientific training is really valuable. So he has extensive clinical experience and extensive clinical training. So he has been a professional nurse and also teaching. So he's been a teacher of nursing for many years, for decades. And he also has a PhD. He got a PhD in international nursing education, teaching bioscience, and he's very passionate about teaching. And I'll add this, that actually he's really good at it. He's really good at making these YouTube videos. He tends to be informative. He's entertaining. He tends to be dispassionate. And he has a certain style where he uses the overhead cam and a fountain pen and sort of walks through papers with the document camera, and I find that I could learn a lot from his style in doing videos, in working in my teaching. So I teach university students, undergraduate and graduate students, and of course I've been doing this remotely. I'm interested in remote teaching, so I'm interested in how he does it. And I think in a lot of ways he's been very good. His area, though, is really at that clinical area. And what I've noticed, and in particular on this video, the video that I'm responding to, is that he tends to get a lot of things wrong. He misunderstands some things when he gets to the cellular and molecular level. And as those misunderstandings turn into mistakes, and those mistakes turn into misinformation, and misinformation is dangerous in a global pandemic. And so that was what prompted my decision to respond to that video with my own video. Now, a day after he posted that video on the Pfizer's new medication and ivermectin, he posted another video because people who had shared his video on, say, Facebook, they had that video flagged as misleading or false information. And he posted a video where he really took down the fact checkers. And part of that was dismissing their qualifications because they were journalists, they weren't scientists, they had no, in the way he described it, they didn't really have the relevant scientific training. So while I'm against arguing from authority, I do think it is worth just putting out my credentials for this so that at least where I'm coming from is clear and where I'm not coming from is clear. So who am I? Why should I have the right to criticize Dr. Campbell, who has such an enormous following and is so good at what he does? My background is not clinical. I've never treated patients. I've been a patient, but I've never treated patients after the Boy Scouts first aid class, I guess. But I do have a PhD. So I did my PhD in molecular biophysics and biochemistry. I got my PhD from Yale University in the early 1990s. I did my postdoctoral fellowship in the Department of Biological Chemistry and Molecular Pharmacology at Harvard Medical School. After that training, I went into biotech. So I had a career, basically the first half of my career, or more, slightly more than that, after my postdoc was in biotechnology and drug discovery research. And that eventually brought me to Singapore. Then I moved back into academia. So I'm currently a professor in the Department of Biological Sciences at the National University of Singapore. And I want to be clear that this video represents me. I'm representing myself as an individual. I'm not representing my employer. The other thing in terms of criticism, because I noticed when I put out a short video about whether I should do this video, I got a lot of people questioning whether I was being paid off to be critical of Dr. Campbell. And the answer is no. I have no conflicts of interest. Not only I don't hold any financial interests in any company with vaccines or developing vaccines or with treatments or developing treatments for COVID-19. I don't do any consulting for any of those companies. And furthermore, my YouTube channel is not monetized. So this video doesn't make any money. I don't make any money from YouTube. And if in the future this channel is monetized, I promise today that this video will be forever excluded from that monetization. So now an outline of this talk. So this video. So I am going to follow along the lines of Dr. Campbell's video. This is the video that I'm referring to where he described the new Pfizer drug and ivermectin. And he says this is a new Pfizer antiviral and ivermectin, a pharmacodynamic analysis. I want to start just clarifying Dr. Campbell's central claim that he makes in this video, and then I will introduce the drugs. So there are 3 drugs of interest. There's Pfizer's new molecule, there's ivermectin itself, and then there's another drug that is part of a co-administration with Pfizer's new molecule in the formulation that Pfizer has in clinical trials. I'll discuss the target, the 3CL main protease from the SARS-CoV-2 virus, how that target works, and how these drugs, or at least how the Pfizer drug interacts with it. And then I'll walk through the claims that Dr. Campbell makes along the way. And while it's not universally mistaken, but there are some mistakes and misinterpretations and misunderstandings. And I'll touch on some of those and then bring that together in why the central claim that Dr. Campbell makes in his video doesn't hold up. So this is the central claim. The central claim for Dr. Campbell in that video is that Pfizer's new antiviral protease inhibitor, PF07321332, uses the same clinical mechanism of action, or he calls it mode of action, as ivermectin in treating COVID-19. That claim does not hold up. But also, I don't want to paraphrase Dr. Campbell's claim, so I think at this point it's good to hear from Dr. Campbell himself.
And that drug works in a particular way. It has a particular, what we call a pharmacodynamic action. It works in a particular way. But there's another generic drug called ivermectin that you might have heard of that works in exactly a same way as that. Now, no one's saying that information has been deliberately suppressed for years while millions of people have died. But what we are going to show on this video is conclusive proof from the literature that this modality of action is the same.
Okay, so there we have it. That's Dr. Campbell's claim in his own words. And I think it should be pretty clear that I'm not misrepresenting Dr. Campbell's claim in this. So let's move forward and talk about the science. So we'll start by discussing the drugs, the 3 molecules at issue here. The first of these is this new Pfizer compound, this new Pfizer antiviral, PF07321332. It's shown up here in the chemical structure, and Dr. Campbell talked about this in his video. I want to focus not just on the overall shape, but just a couple of things about this. So Dr. Campbell focused on these 3 fluorines, this trifluoromethyl part, but actually the business end of the molecule is right here, is this nitrile. That's where the molecule has a covalent interaction with the target, as we'll see. Also, you might notice that elsewhere on this molecule, you see this kind of thing, this peptide. This is a peptide bond. This is another peptide. There's another peptide over here. And so this is what's called a peptidomimetic. It is designed to look like a protein, designed to look like a peptide. And in that sense, that's because that's the substrate of the 3CL main protease of SARS-CoV-2. The other thing is you might notice this weird bicyclic ring here. That ring is to constrain the structure of this molecule so that it fits into the pocket and looks a lot like the protein, even though it's really not. These fluorines here, they bind to some water molecules. Okay, or they hydrogen bond to some water molecules. Okay, so let's move forward to the next compound. So the next compound is ritonavir. So ritonavir is, I'm highlighting this, it was mentioned by Dr. Campbell, is also a protease inhibitor. It's been around a long time, since the mid-1990s, was originally developed for HIV. This is another peptidomimetic. You can see these peptide bonds in various places around this. This is nearly symmetrical. It's sort of more, it's sort of pseudosymmetrical one. And it is, but in this case, it's not actually used as a protease inhibitor. So it's sort of spiked in to join this previous compound in a treatment, in a pill or tablet. And what that does is it slows down the metabolism of this one. And that increases the concentration in the bloodstream of this one. And that's very important. That's called a pharmacokinetic effect. And so this is useful. It's not used very much as a protease inhibitor itself, but it's used in combination because of those pharmacokinetic interactions. And then the third molecule is, of course, ivermectin. So ivermectin, so they're actually, ivermectin has two molecules. They differ way over here on the right side. They differ a little bit. There are these two isomers. This is one of the two isomers. The other one differs a little bit over there. This is actually the minor isomer. It's only less than 20% of the mix. But I put it here because this was the one that Dr. Campbell highlighted in his presentation. You'll notice that this is not a peptidomimetic. It doesn't resemble a peptide at all. It doesn't have any nitrogens in it, just not any fluorines, but it doesn't have any nitrogens as well. This is what's called a macrocyclic lactone. That's because of this part over here and this cycle in this area, the cyclic part in this area. So what ivermectin does, is it binds to glutamate-gated ion channels. Okay, so an ion channel is a protein or complex that allows ions to cross a membrane in an organism. And this is what's called a glutamate-gated ion channel because it requires glutamate to open. So glutamate will open or close it. And so glutamate is used to regulate the opening of that ion channel. And in this case, the main target of ivermectin is a chloride channel, glutamate-gated chloride channel. And one of the reasons that ivermectin is so safe is because we mammals, and I think all vertebrates, don't have any glutamate-gated ion channels outside of the brain and the spinal cord. And ivermectin can't get into the brain or the spinal cord because of the blood-brain barrier. So it can't get to those ones that we have, and so it's safe because the safety of ivermectin is because is at least partly because of that specificity. Whereas these parasites have these glutamate-gated ion channels, and vertebrates do not have them accessible to ivermectin. Okay, now we've covered those drugs, so let's talk about the target, which is this 3CL main protease from SARS-CoV-2. So here's this peptidomimetic covalent, so it has a reversible covalent interaction, as we'll see, with the 3CL protease from SARS-CoV-2. This should say SARS-CoV-2. And here's ritonavir, with which it's administered. So what happens is SARS-CoV-2 comes in through binding to the ACE receptor and comes into a cell. And then the RNA genome is used to translate to these polypeptide chains. So this is not uncommon in these RNA viruses for the proteins to be translated in more than one, and then they have to be cleaved. And that's what the protease is for, to basically cleave to the mature proteins. And so that gets translated, it gets then cleaved by these proteases, and we get the mature proteins, which are then used to create the next generation of viral particles. This is actually a three-dimensional structure from the protein data bank of this of the target itself, the 3CL main protease from SARS-CoV. That was, I think, from SARS-CoV-1, but actually this structure, as we'll see, of SARS-CoV-2 has been solved. So we have a good idea of what that looks like structurally. Now, there's no role yet for ritonavir. So what's ritonavir doing in this? Well, as I mentioned, ritonavir is used in combination with the new agent. And what it does is this all takes place in the liver. So in the liver, there are these proteins, these cytochrome P450, a family of proteins, which basically metabolize lots of drugs. And the most widespread of these is this CYP3A4 family. They're subforms of that. Both PF07321332 and ritonavir are metabolized through the same enzyme in the liver. But ritonavir is metabolized very slowly. And so having ritonavir there basically blocks that enzyme and basically prevents the rapid metabolism of this new agent. And that allows this new agent to stay in the body, in the bloodstream, for a longer period at a higher concentration before being converted to waste. So this type of interaction, this type of pharmacokinetic interaction is really not uncommon. And in fact, if you or anyone you know takes a statin like Lipitor, Zocor, Mavacor, if you take a statin, the statins also run through this same family. And people who are on statins to lower their cholesterol, what they're told, usually by their doctors, is that they should avoid grapefruit juice, pomelo, and pomegranate. And the reason is because grapefruit, pomelo, and pomegranate have a compound in them that is just a natural thing in the juice that is metabolized through the same enzyme, but metabolized very slowly. And so if you drink a big tall glass of grapefruit juice and then take your Lipitor will actually be at a higher concentration than intended. So you should avoid grapefruit juice. You know, the recommendations are to avoid that. And that's because of the pharmacokinetic interaction. So this type of thing is very common. Sometimes it can be the source of adverse events, but in this case, it's being used to kind of regulate ultimately the, ensure that the PF07321332 is available at the right concentrations to have its pharmacodynamic effect. Now, this is a recent paper, which is very interesting. And in this case, these authors have crystallized and solved the three-dimensional structure of the actual target complex with the molecule itself. So here we see that molecule. And what one can see, this very interesting interaction, molecular interaction, is that there's ultimately it forms a covalent bond between this nitrile and this cysteine 145 with the help of this histidine giving up a proton here. And that covalent bond, it's reversible. It's a reversible covalent bond. That's a putative mechanism for this. But that really ties this thing in very tightly, and the covalent bond keeps it there. Here you can see a schematic down on the bottom, and in that schematic you can see these fluorines. One forms a hydrogen bond here, but the other interactions are to these bound water molecules. That's what these W2 and W3 mean. Okay, so that's just a little bit about that target. All right, so that's the background. Let's get into the evidence. The first paper that Dr. Campbell discusses, and he doesn't make any real claims about this paper, but I'm delighted that he mentioned it. So this is a paper of identification of 3CL protease inhibitors by a quantitative high-throughput screening. So this is a group at the National Institutes of Health in the United States, and they do this screen. Now, he points out, Dr. Campbell points out, and he's quite correct, that they go through these several screens. They go through a primary screen, a hit confirmation screen, and a counter screen. The reason they do this, and they use this fluorescence resonance energy transfer to do the detection. The reason that they do this is this first screen is cheaper. It's not cheap, but it's cheaper. And at this point, they're working with over 10,000 compounds. So they do that screen. That's a biochemical screen, just of the inhibition of the enzyme. They take the top results from that, top 161 results from that, and they confirm those. And then they do this from the top results of that. They do a counter screen, which is to just check for any inherent autofluorescence issues from the compounds. So that's the basic idea of the screen, and it's typical this way. I'll put up a link, or where is it? Here. Is it here? Here. Yeah, I'll put up a link there to a video from someone else on high-throughput screening, if you're curious. But one thing about these high-throughput screens, think about this, there's a large number of compounds, 10,755 compounds. So a lot of this is done via automation. So they have these libraries of compounds. You can purchase thousands of compounds in libraries. And when I say a library, what I mean is something that usually comes on this kind of thing, like plates. So this is a plate. This is from my lab, and this it has 96 wells. So it functions like 96 little test tubes. And then there are robots that basically can stamp these out or work just 8 at a time. This is 8 by 12 at a time, and run these reactions. You can get plates with different coatings on the bottom for different types of detection. These are the tools that get used for these kinds of screens. And in fact, in an academic lab like mine, we very typically use 96 well plates, but in a big lab, like the labs that we're talking about here, in a big screening setting, a high throughput screening, what they'll do is divide each of the wells that I have in a 96 by 4 or by 4 again, and can get up to 1,536 wells on a plate of exactly the same size. And of course, this is too small to handle manually, of course, and instead it's all handled by robotic systems to do the screening. Now, the reason I mention all of this is because it helps to understand what the data is. So this is the data from their paper for the very top ones. So these are some of the top ones, and the blue lines here represent the data that was collected in that first screen of over 10,000 compounds. And then the green and the black are the confirmation screens, different things. They're more expensive. They're biological screens, not biochemical screens. And you can see that they give different results. Where the curve turns is where the drug is said to have its action. So that's where it really does its business. But you'll notice that in some cases, there's very little happening. So here we don't even have a curve drawn through these points. Here we don't have a curve drawn through these points. Here we have a curve, but it doesn't look very informative. So we get data like this. It's very common to get data like this where not everything fits. So only the ones that fit get reported, the rest get not reported. And that's very typical because not everything is going to have an effect. Now, the thing is, I looked at the paper. And when I looked at the paper, I looked at what libraries they're using for screening. And they were using libraries for screening that contain a lot of approved drugs and investigational compounds. And so this library is quite large. And since it's from the US NIH, they have an obligation to actually make their results public. And so I wanted to check, is the data available? And it is. And so since this is approved drugs, and investigational drugs, one of these libraries, there are several libraries here, might have ivermectin in it. So maybe they already tested ivermectin in that screening paper. The data is available, so we can go and have a look. And let's do that. Okay, so here we are at the NIH's COVID-19 Open Data Portal, which is where the data is made available. These results are made available. And from there, I have several tabs. I'll put the links all in the description below the video. So here we see this, the National Center for Advancing Translational Sciences. This is their pharmaceutical collection. So this is, their pharmaceutical collection, their molecules that they study, small molecules and so on. And in this, we can look at different types of assays. So that's where it really starts to get interesting. Let me make myself disappear there. So here we have these different SARS-CoV-2 assays. And we don't have to go very far before we see the 3CL enzymatic activity assay here. So that 3CL enzymatic activity assay is here. And this is done in 1536 well plates, so the very big plates. And this is done, I checked, this is basically the same assay. The way they report it for the website, because they're making the assay results, the screening results public, it's a little bit different, but only a little bit different from the way they report it in the paper. It's basically the same. And you can export the screening data and basically export a file that you can then import into Excel. So I've done that. And so here is the Excel file itself. Now that is definitely too small to see, so let me make that a little bit bigger. So here we see these samples and a sample ID and a sample name. I've added a column in the beginning, which is this known to be ivermectin? So sometimes the sample name says ivermectin, and sometimes there's another column over here you see called alias, and alias mentions ivermectin. And there are three, I'll give that away. There are three that say ivermectin. There's another one which is, so there are three that have ivermectin, and then there's sort of one other, it's not an alias, but there were two other molecules that are not ivermectin, but they're often given instead of ivermectin. So there are other antiparasitics that are given instead of ivermectin in some cases, and they were listed there because I think that column lists pharmaceutical alternatives. At any rate, there are only three that are ivermectin. So let's look at the data. So the data has sample names. Let me make that a little bit smaller so it's not so big. It has a PubChem ID, which is a chemical identifier. It has the primary mechanism of action of known to be the drug if it's known, and then the assay names. These are all the same assay names. It tells us the library, so I mentioned that this is from several different libraries, and so on. Now, in the paper, they screened 10,000 compounds. If we look at this, they actually have somewhat more than that now, 12,249 compounds, so it's a little bit more than they had in the paper, because they've screened more compounds since the paper was done. And the results start being over here. So here we have things like AC50, which is related to the EC50 or IC50 that they report in the paper. This is basically the point on the concentration curve where it reaches roughly, basically 50% of the activity. And this is reported in micromolar. It's also reported in as a log base 10. And then there are these other things, efficacy, so closer to minus 100, the better. And then max response is the largest response. So these are various things. As you saw on the previous data, on this kind of data, these curves don't always give us a good fit. So that's a general truism for this type of screening data. So some of these are going to be blank, and that means that they didn't give a very good fit. And so it would be meaningless to talk about, say, an AC50 for this assay for this molecule. I checked because this case, the sample IDs are not always, don't always convert to a common name. So not all of the common names of the top ones show up from the paper show up in the same way here, but those that do show basically the same results. So I've confirmed that the results hold up. This really is basically the same data as from the paper. So what we can do is we can say, okay, let's ask, now if we see here the, where am I looking? Oh, I'm there. If I'm looking, the real thing is the AC50. What is the micromolar concentration where you get 50% of the maximum effect? And so we see that, and I can sort this. Let's sort this from smallest to largest. And one thing we notice down here is that it stops at 453. So we don't get any after 453. And that's because only 453 of the 12,000 molecules tested even gave a curve, so that none of the other ones gave anything like an estimate of AC50. So we get nothing out of those. They might have other measures, but they don't get that sigmoidal curve. Okay, so that's what we have. And let's actually check now which of these. Let's select for the ones. So the ivermectin is either, it's off the edge there. Whoa, let me make that a little bit better. The ivermectin column there is either yes or blank. Okay, so let's just drop the blanks and keep the only yeses, and there are only three. Only three here. Two of them are actually ivermectin, and the third one is this one here that is has an alias of ivermectin, as you can see here. So actually this screen, was screened 3 times. Ivermectin was screened 3 times in this screen that Dr. Campbell pointed out. How did it do? Well, remember, only about 430 even gave an AC50, and the only one of the 3 that gave an AC50 was number 356 in the list. So it was near the bottom of the list. And the other 2, which are also ivermectin, they weren't even able to get a fit. Okay, so here we have 3 data points, and one of them gave a fit at a high AC50. You can see the top AC50s are much lower than this. The other data is also not compelling, so the maximum response is 35 and efficacy of 41. So it only affected about 35 to 41%, and neither of the other ones had any measurable effect. Why is it in 3? Why did they screen it 3 times? It's because these were 3 different libraries, and ivermectin is so common that it was in all 3 libraries. So that's the result of this screen that I'm glad Dr. Campbell pointed us to. So they did actually test it, absolutely. And so here we can summarize the paper. Okay, they have done this quantitative high-throughput screening. This is a very, very strong screening group. I've read a number of their papers. And yeah, they screened, it's actually now more than 12,000 compounds. I mistyped that. They screened more than 10,000 compounds, and now more than 12,000 compounds against SARS-CoV-2 3CL main protease. They actually included ivermectin in this screen 3 different times. I put this as a circle because that wasn't noticed by Dr. Campbell, but this check mark is an indication that, yes, we agree. Dr. Campbell and I agree on this. This part here is something that he missed, but the last one is ivermectin didn't show evidence, and certainly not consistent evidence, of 3CL protease inhibition in this screen. And this is a very good group. They developed the screen specifically for this purpose. They're world leaders at this. Now, they did get the 1, right? So they have this 1 with an AC50 of around 39, according to this data, similar in the but it's not reported in the paper because it was at the bottom of the list. And we can come back to that a little bit later in this video and relate it to the results of another paper that Dr. Campbell points out. Okay, so that's the end of that first paper. Let's go to the second paper. This second paper and Dr. Campbell's interpretation of the results of the second paper. This second paper was published in Physical Chemistry and Chemical Physics, and the title is Microscopic Interactions Between Ivermectin and Key Human and Viral Proteins Involved in SARS-CoV-2 Infection. Dr. Campbell makes a claim regarding this paper. He says, and I'm quoting here. So it's telling us how ivermectin is working to block the activity of the 3CL protease. That's what that paper is about, and it makes perfect sense if you do take a bit of time to read it. I would like to allow Dr. Campbell to speak for himself, so I'll have a little excerpt or two from his video.
Next, there was a lot of papers on analysing these. So this is microscopic interactions between ivermectin and key human and viral proteins involved in SARS coronavirus 2 infection. Now, obviously, you want the evidence for this. You're not going to take my word for it. Here it is. It's on this paper here from no less than the Royal Society of Chemistry. And again, thankfully, all these are available. And if you're a biochemist, you'll understand that. If not, you'll just get the gist the same as me.
Okay, so here I want to pause because I think it's pretty clear, and it's clear from later interpretations of the paper, that Dr. Campbell has missed something absolutely critical in this paper. This paper is entirely in silico. There's no experimental data. They never touch ivermectin. They never touch any of these proteins. This is computer simulations entirely, and it's computer simulations of possible interactions between ivermectin and 4 possible protein targets, including the 3CL main protease. The other ones include the ACE2 and RBD, which are the targets of the virus itself, and then these are the 3 viral proteins. So this is entirely an in silico paper. There is 0 experimental data or evidence presented in this paper.
But I think we have got the gist. Now, that's the reference for that one there. The strength and persistence of the interaction between ivermectin and the binding site of the 3CL protease indicate that a partial inhibition of the catalytic activity, in other words, the way that the enzyme works, could have a place as the drug interactions with the main subdomain that define the enzyme define the enzyme binding pocket. Now, the enzyme binding pocket, of course, the protease is an enzyme.
So at this point in the recording, Dr. Campbell spends a little time just clarifying what is meant by an enzyme binding pocket. But I want to pause here because I want to emphasize, I want to just point out here what Dr. Campbell quoted. This is the part that Dr. Campbell quoted, and it seems very compelling. It ends here with a colon. And the reason is because Dr. Campbell left off the end of the sentence. So the full quote, this is from the paper itself. Here's the part that he quoted. Hence, the strength and persistency of the interaction between ivermectin and the binding site of 3CL Pro indicate that a partial inhibition of the catalytic activity could have place as the drug interacts with the main subdomains that define the enzyme binding pocket. Then this colon here, and it lists basically the subdomains. So it makes sense maybe not to quote that part. But the part here at the end is critical because it ends after that, although with a strong dependence on the initial conditions of the trajectory. So what is the trajectory? The trajectory is the conditions, the setup for the simulation. So if you start the simulation and you orient ivermectin one way and the protein another way, it won't work. If you start in another orientation, it might. In another orientation, it might. In another orientation, it might not. So it's very dependent on how the simulation is set up, whether it works at all in this simulation. Again, there's no experimental data, 0 experimental data. It's entirely simulation. But in that simulation, they put this big caveat, which unfortunately, Dr. Campbell didn't quote. The authors of this paper have quite a bit more to say about 3CL protease. Remember, they did simulations against 3CL protease and against 3 other proteins, 2 other SARS-CoV-2 proteins, as well as the ACE2 receptor. So what do they say about 3CL? They say here, the proteins considered in this work are not established targets of IVE, ivermectin, since it has been suggested that the main antiviral action of ivermectin is deployed through the inhibition of important A/B1 or alpha/beta1 carriers. That's at least more consistent with ivermectin's main target as a glutamate-acted chloride channel. Then they go on. Consequently, no biological or clinical outcomes of possible anti-SARS-CoV-2 activity of ivermectin can be explained only from the present data, since they will also require or to take into account the balance with cellular functioning and bioavailability. I actually think that's even optimistic. There's no experimental data here. This is all computational. I don't have any objection to computational data, but one has to recognize its limitations. More concisely, they're advocating don't do what Dr. Campbell is doing, which is saying that this is kind of an explanation for how, for an effect, a biological effect of ivermectin on SARS-CoV-2. Don't do that, they're saying, and they're doing it. Now, that's in the text. Of course, Dr. Campbell also quoted within the text. There's a part that I was really surprised that he missed, which is in the abstract itself, right? So the very opening of the paper, the last sentence of the abstract says, results reveal, so this is again in silico, results reveal that the ACE2 protein and the ACE2RBD aggregates form the most persistent interactions with ivermectin, while the binding with the remaining viral proteins is more limited and unspecific. So what they're saying is that to the extent that this in silico work is meaningful, it is, they want you to interpret it with respect to ACE2 and the ACE2-RBD complex. They don't want you to interpret it with respect to any of the viral proteins, and that includes the 3CL protease. And the reason is because even their simulation data doesn't, doesn't say enough about it. Now, let's just pause here and let Dr. Campbell sum things up.
So it's telling us how this is working to block the activity of the 3CL protease. That's what that paper's about, and it makes perfect sense if you do take a bit of time to read it. So
let's summarize what we have here in this paper that we've just gone through. This is an in silico study, entirely in silico. It's entirely simulation of possible molecular interactions between ivermectin and actually 4 proteins, including 3CL protease is one. There's no experimental data. There's no positive or negative controls or calibration. In this kind of thing, one has, it's a good thing scientifically to ask yourself, what would a negative result look like? What would a negative result look like? And you know what? It would look exactly like this. It would look exactly like this. That is to say, there's no, when you do a simulation like this and you ask these 2 molecules to interact, somehow they will, you'll probably get some sort of interaction, but whether that's meaningful or not depends on a lot else. One thing that they could have done is they could have included some positive controls, like ivermectin. If it's possible to model, I don't know enough about whether the ion channel target of ivermectin's structure has been solved, but if that has been solved, they could do something with that. They could do a known inhibitor of the 3CL protease. There are lots of ways to do this to get some sort of positive or negative controls or calibration. In this case, it's a bit of a fishing expedition, and of course they found some things. So that first part was missed. Dr. Campbell missed that, which is why it has an open circle. He took this quoted passage out of context. I'm a little sad about that. I don't know that it was intentional, because I think he missed the in silico nature of the paper, but also because that the clause which had that major caveat was after this list of these domains. Now, the authors say the results should not be used to interpret the biological activity of ivermectin. And the authors claim that the simulations of the ivermectin 3CL protease interaction are limited and unspecific. So whatever this paper says, it does not clearly say about basically a bunch of interactions and a mechanism for ivermectin inhibiting the 3CL protease. It certainly does not say that. Okay, so the next paper in this series is this paper from Communications Biology, which is a Nature journal. And this paper is titled Identification of 3 Chymotrypsin-like Protease, 3CL-Pro inhibitors as potential anti-SARS-CoV-2 agents. And this is a mix of an in silico and experimental paper, and I would say it's the strongest of the papers that Dr. Campbell looked at. And so why don't we just let him introduce it?
Now, the next paper is this one, again giving you all the evidence, identification of 3 chymotrypsin-like protease inhibitors as potential anti-SARS coronavirus 2 agents. Don't worry if you're not getting all the terminology, I don't always get it myself, but that's this paper here. Out of 13 OTDs. Now, OTDs, this is called off-target drugs. So ivermectin wasn't originally one of the antivirals they were looking at. They were looking at other existing antivirals. So that's why they're calling it an off-target drug there.
Okay, I think at this point, it's a good time to just review a little bit about what drug target and what off-target means. And so for this, I'm going to switch to the DocCam. Okay, so here's my DocCam. And so what I've drawn here is, well, this is a target. This is a drug target with my attempt at drawing, which is quite poor. And I've drawn this at this point here. So that's the target, okay? And then I've drawn over here on the y-axis of this a drug with this square braces, meaning the drug concentration. So the drug concentration is going to get higher as we go up this. And so what does this x-axis mean? Well, this is a little bit abstract. So here's the target, and these are sort of off target. So anywhere to the left or the right of this is off target, and the target is there. Now, typically, what happens is that the idea of the target is that the drug is specific to that target. So wherever the drug requires the least amount of drug to hit, that's the target. And so off target generally requires raising the concentration. So it kind of looks, again, this is a little bit abstract, but it kind of looks something like maybe it looks something like this. So the drug target, you only need a little bit of drug, whatever this concentration is. And then as we get off and we start to hit other targets, we would need more drug. Another way of thinking about this is that as we raise the concentration of the drug, we hit more things. So we get further and further off the target. And so sometimes this is beneficial, sometimes it's not beneficial, and then sometimes in these higher kind of off-target areas, we get into toxicity. So just to make sure, because my drawing isn't very good, I want to make sure that our UK friends can recognize this is a target. So I have my old dart here. It's a little worn, but this is the target. That's the bullseye. Everything off of this is going to require a higher concentration of drug. So when we talk about off-target effects, we're generally expecting something like this, some sort of higher concentration of drug to have an effect. Okay, so let's get back to this paper. What did they do and what are their claims? So this is the work. It was also, much of it was computational modeling, but not all. They started with a computational model, an in silico screen of a fairly large number of compounds. Then they did a biochemical screen on the top hits. Unlike the previous paper, they didn't do any confirmatory screens. They didn't do any biological screens. They didn't do any significant counter screens, although they did check for autofluorescence issues. And then they did some more detailed computational modeling on the top hits. The paper's main positive claim is that ivermectin, alone among the approved agents, blocked at a high level the 3CL protease. So they say it blocked 80% at 50 micromolar concentration. And they claim that then it has potential for repurposing. Now, so this is the figure that they showed to that effect. And so here's ivermectin. Let's see if I can, yep, there we are. Okay, so here's ivermectin here. And the short bar on that bar chart means that it is being inhibited. This is so they did some nice controls. This is a negative control. And this is a positive control that they purchased along with the commercial assays that they got from the company. So unlike the earlier paper, they didn't develop the assay. They're not kind of at this. I'm sure they have done assays before, but they're using a commercial assay. They weren't kind of innovating on the assay itself. So those are their major positive claims. And so one thing we can do is we can go back and say, okay, let's accept the claims. Let's say, okay, let's accept all the claims about this first bullet here. And let's ask ourselves what that means for ivermectin repurposing, for actually using ivermectin, if this claim is true, to block 3CL protease. And so this is the actual data from the paper itself. Actually, this is not a figure that Dr. Campbell presented, but I think it's a really nice figure. And this is, in fact, maybe the best paper in support of the claims made. So here we see ivermectin, and you see this inhibition is this curve, and you can see that it happens kind of around in the 20 micromolar area. These are units on a log scale in micromolar. And so we have a sense that 20 micromolar is when this thing kind of crosses 50. So that's where we get the IC50 of 20 micromolar. And you can see these other drugs don't seem to do as well. Okay, so we have that value, somewhere between 20 and 50 micromolar, 20 for 50, 50 for over 80% blocking. So that's good, okay? So what about ivermectin's actual target, the target that ivermectin is designed against? Well, that target is this glutamate-gated chloride channel. And so this is an assay for that. This is actually a biological assay, so it's a little bit harder than this, but this is the binding of ivermectin to C. elegans membranes where these channels are. So it gives us a good indication in a biological condition. So this is kind of an upper bound on that. And we see a KD, this is the major measurement here, a KD of 0.26 nanomolar. So that gives us a rough idea that it's less than 1 nanomolar is the level of target inhibition, or the level of ivermectin that's required to inhibit the target at 50%. So let's go back here, and now we can draw a number for this point down at the bottom here. And let's say ivermectin is not as good as it says it is. So let's put this at 1 nanomolar. And let's use these ticks as a log scale, because we're going to be using several orders of magnitude. So this is 10 nanomolar. This is 100. This is 1 micromolar, 10 micromolar, and 100 micromolar. Okay, so those are some concentrations that we can use. And we can say, okay, let's use green for the target. So we know our target is going to be hitting really here at this one there, and our what we are trying to achieve in inhibiting the 3CL protease is going to be something in the neighborhood of 20 to 50. So let's aim at 20. I think that's kind of a rough log scale of 20. So we want to get somewhere about here, about this level. Okay, this is our goal. And this is where we are in terms of the actual target. So let's ask some other questions. So what can we say about some of the off-target effects? And so there are a bunch of papers. So the paper that this paper here is an older paper, but it's a relevant paper on mode of action of ivermectin, and it includes some off-targets, and then there are other more recent off-targets that are cited in the literature. So going with this paper from a year ago, this is on pharmacokinetic considerations for repurposing ivermectin for treatment of COVID-19. And now the pharmacokinetics of ivermectin are very, very well understood, as well as the pharmacodynamics to some of the other targets, some of the other ion channels that ivermectin is known to target at somewhat higher levels. Okay, so let's go back to this and start to fill in some of those blanks. So we know that the glutamate activated glutamate-gated chloride channel is around 1 nanomolar or less. Other ligand-gated ion channels are something on the order of 100 nanomolar, like maybe something like that, maybe 100 nanomolar to, so they're somewhere in this kind of other, so this would be something like GABA channel, other ligand-gated, maybe I think the glycine channel, glycine receptor is somewhere in this ballpark. Okay, so those are possible. Biochemically, those are possible. And then the 3CL protease of SARS-CoV-2 is here. Okay, so this is kind of where the range that we're looking at. Remember, this is a log scale. So maybe it's achievable. We just have to raise the concentration of the drug about 20,000 to 50,000 times the concentration that it needs to hit the target in the intended target, the glutamate-gated chlorine channel. Is that possible? Can we actually reach this kind of level? Okay, so there we can turn to the pharmacokinetics. So it's known, and this is from references in this paper, which I'll put a link to. It's known, the pharmacokinetics are very, very well known for ivermectin, so we can basically fill those in. And so if we fill those in, we can ask if ivermectin is given in the blood, and it's given, we just keep increasing the dose, what's the maximum level that we can reach? And in fact, actually, that's a figure on this paper. It's right here. This is actually not quite it. So let me fill in the parts that make this more sensible. So the maximum plasma levels are about 0.28 micromolar. So that's about something like this. This is our, let me use a different color if I can get a different color. Okay, so here's a blue. I'll put this in a dotted line. So this will be about 0. I'm going to try to estimate a log level. So this is something like this. 0.28 micromolar is basically max plasma. Okay, so that's our max plasma concentration. That is, and that's actually at about 8.5 times the FDA approved dose of ivermectin. This is about how much ivermectin would be in the plasma, in human plasma, if we were giving 8.5 times the amount of the FDA approved dose. But it's a little harder than that, because it turns out that ivermectin, most of ivermectin, about 93% of ivermectin, is bound to plasma protein. So actually only 7% of ivermectin is free in the plasma to be taken up by cells. Let's say 10%. Let's be generous here, and let's say 10%. So that puts us something more like this, which is going to be the max free plasma. Oh, I get my face out of there. The max free IVM concentration in plasma. Okay, so if this is 0.28, this would be 0.028. So we're something on the order of 1,000-fold less than we need to be. And that's assuming that the biochemistry is replicable completely in the cell, which is not usually the case in drug development. Okay, so that's sort of where we are with this. And so what does that mean? Well, so that means if we summarize the results of this paper, we can, for the sake of argument, we can accept every claim in the paper at face value. Every claim. Just say, okay, sure. Maybe they're exactly right. Ivermectin inhibits the 3CL protease at 80% at 50 micromolar. And that means achieving the biochemical effect would require something like 20 to 50 micromolar. And it has to be either in the blood or it has to be in the applicable cells, like the lung cells. The maximum free ivermectin in plasma is something like 0.02 micromolar. So a lot less, right? Thousandfold less. And we don't even know the max in lungs. So the best model for a max in lungs would be based on injected ivermectin in calves, and that would put an estimate of about 0.1 micromolar. And in addition, this paper has an additional caveat, which is that the detailed molecular modeling, which I mentioned here, this more detailed computational modeling, one of the results of that is that they aren't able to get a good computational model for ivermectin binding to the 3CL protease unless it's a dimer. So the protease is a monomer or a dimer, and only when it's a dimer does ivermectin stably, even in a computational model, seem to bind. Accepting everything, that's basically the best one could get. Now, so that would mean that if ivermectin did everything that it's claimed to do in this paper, which is actually the strongest paper, the only paper that presented real experimental evidence in support of this idea, if ivermectin did everything it's claimed to do for the 3CL protease in SARS-CoV-2, we wouldn't be anywhere near. We'd be 3 orders of magnitude away from the concentration, 3 to 4 orders of magnitude away from the concentration we need. And there would need to be some total reformulation, like an inhaler or something. So that's where we are with this paper, and that ends paper 3.
Right, next paper. Next line of evidence here is from this paper here. Again, Royal Society of Chemistry, no less.
So I want to pause here because this is the second time that Dr. Campbell has used this same turn of phrase, Royal Society of Chemistry, no less. In both cases, the first one was in Chemical Physics and Physical Chemistry. This is in a journal called RSC Advances. The Royal Society of Chemistry is the publisher of those journals, but that doesn't represent an endorsement from the Royal Society of Chemistry. It doesn't represent any sort of stamp. It's the publisher, it's not the journal, and it's not the author. In this case, the journal is a journal called RSC Advances, and their appeal is that they have a time from submission to first decision of 26 days, which is pretty fast by scientific terms, actually very fast by scientific terms, so it's quite appealing in that respect. But of course, that means from submission to review to decision in 26 days. There's a downside to that as well.
Let me give you the gist of what it's saying. From the docking analysis, that's the way that the drug docks into the enzymes, the way that the drug binds up the scissors in this case. From the docking analysis, ivermectin showed the highest docking score with an average energy of -8.5 kilocalories per mole. I don't know the units. I don't pretend to know it, but it means it bound on quite tightly. Among all compounds, so it showed the highest docking score. In other words, it cluttered up the enzyme. It cluttered up the 3CL enzymes and stopped it working better than all of the other compounds that they looked at. Remdesivir showed the lowest binding energy and highest docking score. So remdesivir doesn't work very well now on this particular analysis anyway.
Okay, there's a lot going on here, and I want to break it down because this is the first paper that Dr. Campbell has really got entirely wrong. End-to-end wrong. So this paper is, it's not about ivermectin. It's also not about 3CL protease. It's not about the target. It's not about ivermectin. It's about this marine sponge metabolite. So that's one thing. The second thing is, this is entirely a computational paper. There's no experimental data in this at all, except for computational experiments. This is a paper where they look at this marine sponge metabolite and look at computational simulations of potential interactions between a model of that molecule and a model of each of 9 proteins in SARS-CoV-2, including 3CL ligase. Ivermectin is thrown in there because it's in comparison with suggested COVID-19 drugs.
From the docking analysis, that's the way that the drug docks into the enzymes, the way that the drug binds up the scissors in this case.
No. The docking analysis is a computational simulation, a computational model of a potential interaction. If it was known that the drug did bind in this particular area, the docking might help us understand exactly how that happened. If there's no data, it's just a simulation of something that may or may not happen. In this case, it's a bunch of different small molecules simulated against a bunch of 9 different proteins.
From the docking analysis, ivermectin showed the highest docking score with an average energy of minus 8.5 kilocalories per mole. I don't know the units, I don't pretend to know it. But it means it bound on quite tightly among all compounds. So it showed the highest docking score. In other words, it cluttered up the enzyme. It cluttered up the 3CL enzymes and stopped it working better than all of the other compounds that they looked at.
Again, no, that's not what that means at all. In this case, the minus 8.5 is actually not ivermectin with 3CL protease, but it's the average of ivermectin, the binding energies. This is, again, calculated from simulation, so there are no experimental data. They didn't measure ivermectin or any other drug binding to any of these proteins in any sort of laboratory experiment. This is entirely from computational simulations. And that refers not to the 3CL protease, but to the average binding energy across all of these. And so in that case, what I've done is I've created a table. So this is now a table. Let me get rid of my own face so you can see that. So this is the table. This is now the average. This here is the 8.5 that they're referring to in the abstract. That's the average calculated binding energy from simulations of ivermectin across all of these. And in this case, a negative number is meant to be a stronger binding energy. So a higher docking score is a negative number. That's a little counterintuitive, but I guess they were expecting that the readers of the article would know that.
Remdesivir showed the lowest binding energy and highest docking score. So remdesivir doesn't work very well now on this particular analysis anyway.
So this is exactly backwards. And let's go to the table once again. We don't need the averages in this case because you can see that remdesivir here has this score of minus 9.9, which means actually remdesivir was, again, according to these calculations, according to these simulations, remdesivir has the actual absolute best score among all of the drugs, among all of the proteins in this analysis, exactly the opposite of Dr. Campbell's interpretation of this data. Now, it might seem like I'm nitpicking and making a lot out of a few misunderstandings, but let's see what Dr. Campbell does with this misunderstanding.
So that's the link for that from the National Institute for Health and Care Excellence. So pretty expensive drug. I believe in the United States, the course costs about $3,000 for a 4 or 5 day course? Can't remember. Anyway, really expensive. And of course, we know ivermectin is available through the World Health Organization at 6 cents. 6 cents. 6 cents, about 5 English pennies a tablet. So it's a bit cheaper. It's a bit cheaper. And from this data, higher docking score, higher cluttering up ability of the enzyme.
So we can see that Dr. Campbell's basic misunderstanding of that data, which is pretty comprehensive, how badly he misunderstands and misinterprets that paper, has basically led him to then promote a bunch of misinformation about, in this case, even interpreting that data about remdesivir and about ivermectin. Now, of course, I'm not defending the price of remdesivir. And of course, ivermectin is cheap. But this is not at all what that data is saying. That data has absolutely no relevance to remdesivir ivermectin question. And it is just really, I think, pretty sloppy of Dr. Campbell to misinterpret that. Even if he didn't understand the technicalities, I think it's incumbent upon a reader of a paper to identify what they don't understand and to read the tables and the figures and understand how what the sentences actually mean. So where are we with this, the second to last paper? In this case, this paper, there's a lot wrong in this. So this is not a paper about ivermectin. It's also not a paper that presents any experimental data, any laboratory data. It's entirely computational simulations. The paper doesn't provide any information about ivermectin interactions with SARS-CoV-2 proteins. There's no data about that. There are a few simulations, but they're really not the focus of the paper, and the paper is really focused elsewhere. Dr. Campbell misunderstood the basic ideas of the paper, the basic results of the paper. He misinterpreted the numerical results. He got them exactly backwards, so that in the one case where he has a comparison between remdesivir and ivermectin, he got those results backwards. And then he turned those misinterpretations into misinformation related to the price of remdesivir and ivermectin, which sort of adds to conspiratorial thinking about people hiding information about ivermectin, which is really irresponsible, scientifically irresponsible for somebody in his position with his influence to be doing. Okay, if you've stuck this far, we are now at the 5th and last paper. Thanks for sticking it out. And I'll let Dr. Campbell introduce this one.
This is from Future Virology. So this evidence comes from exploring the binding efficacy of ivermectin against key proteins of SARS coronavirus 2, pathogenesis, and in silico approach. Now this is very interesting. In silico means this is computer modeled. Now this is incredibly clever and way beyond my ability. It's computerized molecular modeling, but it's a very well recognized science these days.
So Dr. Campbell is correct. This is an entirely in silico paper, and it's good that he recognized it. However, it's the third of the 5 papers that he's presented that is an entirely in silico paper. It seems to be the first one that he's recognized is an in silico paper, but it's the third paper that is an in silico paper. So one of the reasons he got so much wrong in paper 4 was because he didn't realize it was an in silico paper, and so wasn't really able to read it and understand it, I think. And the same is true for paper 2. But this is also an in silico paper, and it's a lot like those 2 other papers in some respects.
So we've documented an intense binding effect of both ivermectin and this to the main protease with a high binding energy is all that means. In other words, the ivermectin well and truly clogs up the 3CL protease, stops it from cutting up the long protein chains into smaller protein chains, therefore it stops the viruses being made.
Okay, this is once again not true, not the case. There's no way to say well and truly to describe an interaction that is basically explored entirely through simulations with no experimental data, no experimental evidence to back it up. In addition, they simulated a bunch of other things, and those other things would suggest that if they were also true, that ivermectin is, and these other agents are basically just pounding around at a lot of the proteins in the cell and with the virus. Again, no experimental evidence to back that up, no comparison with experimental evidence to support those cases, and it's a speculation. And like the earlier simulation models, there's no calibration or positive or negative controls that allow one to say, how strong is this? What would a negative result look like? What would a positive result look like? So it's, again, a bit of a fishing expedition. And so it's perhaps food for thought if you wanted to do some experiments, but it doesn't replace experiments, and it doesn't tell us what's actually happening because there's no actual data to support it. It's simulations. So let's give Dr. Campbell an opportunity to come back to his main argument before I close out this video.
So just as a reminder, given that with this evidence, this was from the original Pfizer paper, it is working this way because the original Pfizer paper says that the new Pfizer medication is designed to block the activity of the SARS coronavirus 2, 3 chymotrypsin-like protease. In other words, it acts the same way as the sellotape round the pair of scissors, as indeed we have given evidence for ivermectin doing.
But have they? Dr. Campbell says they've presented evidence that ivermectin operates against COVID by inhibiting the 3CL protease. Let's look at the evidence that Dr. Campbell presented, or the papers on which he bases those claims. Just to summarize, he's presented 5 papers. The first of those papers uses quantitative high-throughput screening to identify 3CL protease inhibitors. They turned out they included ivermectin in their screens. This is not something Dr. Campbell noticed, but it is the case, and one can look at the actual results. They included ivermectin 3 times, and it did not give a positive result. So ivermectin did not show up as a 3CL protease inhibitor in that first set of quantitative screens. The second paper is an exploratory, entirely in silico study. It doesn't support an ivermectin mechanism of action through 3CL protease inhibition. And in particular, the authors warn against the kind of misinterpretations and conclusions that Dr. Campbell makes and wants to draw. The third study is, in my view, the strongest study providing evidence that can be said to be in support of this. And that's a mixed in silico study and a biochemical screen. The biochemical screen showed that ivermectin could inhibit 3CL protease at somewhere between 20 and 50 micromolar. This is in conflict with the first screen, but it's not, sometimes results conflict, so it's not impossible. However, this is actually a very high result. This wouldn't be considered a very strong screening result because one really wants to get down to low micromolar or sub-micromolar screening for any real medical screen. And in particular, it's much, much higher. It's 3 or 4 orders of magnitude higher than ivermectin's concentration when it inhibits its primary target as an antiparasitic. It's also several orders of magnitude higher than the highest possible human exposure of ivermectin that's ever been measured. And so it's very, very unlikely. It's certainly not the case that anybody taking ivermectin now would have anything remotely approaching that level of exposure. And it's quite unlikely that it would be even possible to reach that level of exposure. And one doesn't know at all the safety profile if one were able to reach that level of exposure. The fourth paper is another exploratory in silico study. That paper is not about ivermectin or about 3CL protease. It's really about this natural product, and they happen to use ivermectin and several proteins from the virus as a basis of comparison. But this study was completely misinterpreted by Dr. Campbell. He misinterpreted it both qualitatively and quantitatively. And I don't think any more needs to be said about that. And the fifth paper, yet another, the third totally exploratory, totally in silico study, it makes some claims about ivermectin and 3CL protease. It also makes some claims about ivermectin and some other viral proteins. It makes stronger claims about those. Again, no supporting experimental data, none at all. So this is, I think, a pretty weak study. It's weak in a lot of ways, a lot of technical ways that I haven't gone into, but it's a pretty weak study. And overall, the evidence that Dr. Campbell presents in support of this claim just doesn't hold up. There's just not that kind of evidence to make that kind of claim. And I think it's irresponsible to make a claim like this, which undermines the value of a really important, potentially game-changing, as Dr. Campbell said in his initial video on the Pfizer compound. He described it as potentially game-changing, and I agree. But then to come back the next day and undermine that with basically a bunch of misinformation about a comparison to an existing drug, a spurious comparison to an existing drug that just feeds conspiracy theories, fuels conspiratorial ideas for which there's no scientific basis. There's no scientific basis to claim that ivermectin is providing benefit to COVID patients through inhibition of 3CL protease, and I will stand by that. And I invite Dr. Campbell to contradict me if he believes that I've misinterpreted any of these papers. Okay, that is it. Thank you if you've taken the time to get through this video this length. It's certainly taken me a while to make it. And I did it because I felt compelled to do so, to try to address some of the misinformation that is getting out there. I think it's important. I think expertise in these different areas is important. I commend Dr. Campbell on a lot of his videos in other areas. I think he's an excellent teacher in a lot of ways, but I think anybody with any specialized expertise, there's a temptation to get out over their skis when they're in an area that is outside of that area. Okay, thanks very much, and maybe I'll see you in the next video.